Now, I've requested your recording, but I requested permission to record. They press that button.
No.
See how that goes. When
Right.
Graeme gets on,
Yeah.
you're going to do that.
Will I make you a code? Oh,
No, you're
thank
going to
you.
make.
So I'm going to allow you to record.
Okay.
There you go.
And then when Graeme gets on, you're going to do the same thing that you did then and just go to put his name and go
I
make.
should put myself on mute. Hi, Graeme. This is Marge here. We'll take that away.
Hello, Marge. How are you going?
Good, thanks. I just wanted to test your audio.
Yeah, so you can hear me clearly?
Yeah, I can hear you clearly. Thank you.
And you can see the screen?
I can see the screen. So you're on co-host. So,
Excellent.
Looks like we're all ready to go. We're just going to change rooms. So that all looks good. There are some people online already. Welcome, everybody. We're just doing a test. And we'll be starting in about 10 minutes. Okay, Graeme, thanks very much.
Thanks, Marge.
We'll talk soon. I'll do the introduction soon.
No problem.
Okay, thanks very much.
Thank you.
You hear it.
It's mute my audio.
Hello, everyone. This is Marge Overs, and I think we'll get this webinar started now. I'm the Knowledge and Translation Broker for the Centre for Air Pollution, Energy and Health Research, or CAR. Welcome to this seminar with Professor Graeme Zosky, and it's part of the CAR seminar series. Today, we are very pleased to co-host this seminar with Kazance, and thanks to Jacinta Shen from Kazance for promoting the seminar through their networks. Today, Graeme will present an overview of the mechanisms underlying the health effects of exposure to air pollution, with a focus on particulate matter. In short, how can breathing in particulate matter lead to illness? Graeme is a chief investigator with CAR. He is a biomedical researcher and deputy director of the Menzies Institute for Medical Research in Hobart. His research program is focused on the role of environmental exposures in determining lung growth and how this affects health and disease. Graeme's presentation will go for about 40 minutes, leaving time for questions at the end. If you have a question for Graeme, please just type it into the chat box. I'll hand over to Graeme now.
Thanks very much, Marge. It's my pleasure to be speaking to you today, and thank you for taking the time to join me. As Marge outlined, what I'm going to try to do today is to give a brief overview of particulate matter from the point of inhalation to interaction with tissue and cells, and how that might be related to or linked to adverse health outcomes. So the first half of the first third really of the talk will primarily be an overview of particulate matter and the health effects. And then I'll move on to talking about how variations in the characteristics of particulate matter, the physical characteristics or the chemical characteristics, can modify the response with some sort of case studies and experimental examples from my lab and from others. So let's get this underway. Hopefully everyone can see my screen. Okay. So I want to start by sort of giving you an overview of how I think about the response to air pollution. And in my research, I try to think about it from 3 points of view. The first point of view is the pollutant composition and how that might impact on the response. So as I'm sure you're all aware, air pollution primarily has two components, a gaseous component and a particulate component. For the purposes of the talk today, I'm pretty much going to focus on the particulate component and ignore the gaseous aspect. So immediately when you are trying to understand the health effects of air pollution, there are several characteristics of the pollution itself that can impact on the response. So you've got the gaseous versus the particulate components. Within the particulate component, you've got various size categories which have varying health effects primarily because they impact on where the particles are deposited when they're inhaled. And then within that particulate matter, you've got variations in the chemical composition. So for example, here in this little inset is example of 2 samples of PM2.5 showing how the chemical composition of that PM2.5 can vary considerably. And so pollutant composition has a big impact on the response. And when considering how pollutant composition can impact on the response, the other critical aspect with any sort of toxicological understanding or toxicological situation is I'm trying to understand the dose. And so when we're thinking about the response to air pollution, are we thinking about an acute exposure event, like an acute combustion or landscape fire here on the left? Are we talking about chronic exposure in a high pollution environment such as this in the middle? Or in what I think is quite an interesting situation, are we talking about an acute on chronic response? So here is a picture of the skyline of London showing Saharan desert dust blowing over the city, which the residents will be exposed to on the background of chronic urban-derived particulates. On top of that, when we're considering the response to air pollution, the other thing to consider is the subject. So are we talking about the response to air pollution in a healthy individual, which might vary depending on whether they're young or whether they're adults? Are we talking about the response in someone who has an existing chronic disease, such as asthma or COPD? Or are we talking about someone who might have an acute disease, such as a respiratory infection, influenza, for example? So when considering the response to air pollution in general, it's the interaction of all 3 of these things that dictate the response. So what I'm going to try to do is to unpack some of these aspects, not all of them, but some of them, to try and highlight how the adverse health impacts that are driven by exposure to air pollution, primarily particulate matter, can impact on the response on the basis of these things. So what evidence is there? So there's lots of evidence out in the literature, and the literature continues to grow at an exponential rate, showing a strong relationship between exposure to air pollution and adverse health outcomes. So the most well-established aspects of the literature are primarily in respiratory disease and cardiovascular disease. So exposure to air pollution is associated with increased morbidity and mortality, both respiratory and cardiovascular disease. More recently, there's a number of studies in a range of other areas that suggest that air pollution may also be related to metabolic disease, neurological disease, ear disease, and kidney disease. So there's a range of health effects that are pretty much spread across the body. So what I'm going to try to talk about is how air pollution, in particular particulate matter, can impact potentially on these diseases, mostly through conserved pathways. So in thinking about the health effects of particulates, the first thing to try to understand is what happens to particles when you inhale them. And the short answer is it's quite complex. So if you think about particles of different sizes that might be in the ambient air, so we inhale them, and at the moment we're assuming that they manage to make it past the upper airway defenses. So we're talking about particles smaller than 10 microns in size. But if you look at the particles below that size, what you find is that the deposition of those particles within the lung varies considerably depending on the size of the particles. So this is quite an informative figure, so I'll try to walk you through it. So here we have particle diameter on the x-axis, so 10 microns or less. So they're the particles we're interested in because they bypass the upper airways. If you break them down into different size fractions, the larger particles will tend to deposit in the lung as a result of sedimentation processes. So these are particles that are big enough such that when you inhale them, they will deposit directly onto the tissue. At the other end of the spectrum, the very small particles, edging towards the nano-sized particles, will tend to deposit on the tissue by a process of diffusion. So they tend to act a lot more like a gas, if you like. The interesting thing is that there's a sweet spot in the middle where there's a range of particle sizes which seem to be somewhere between 0.1 and 1 micron that don't readily deposit in the lung. And that's because they're too small to be deposited by sedimentation processes, but too large to be deposited via diffusion. So you end up with this interesting bimodal pattern of distribution where you have large particles depositing in the lungs and small particles depositing in the lungs, and the middle-sized particles tending to enter the lungs and then they're breathed out with exhalation. On top of that, what tends to happen, as you would expect, is that the bigger particles tend to deposit in the upper parts of the airway tree, and the smaller particles tend to deposit in the gas exchanging units of the lung. So what that means is that depending on the particle size that you're inhaling, it will have an effect on where it's distributed, where it's deposited within the lung, and therefore the health effects, because the types of cells and the types of potential health effects will vary depending on which part of the lung is affected. On top of that, and unfortunately, I apologise for the small figure here, what we know is that pre-existing lung disease will affect where the particles are deposited. So this doesn't project particularly well, but this is a simulation of particle deposition within an airway, and the colour codes on there are different simulation for different severities of disease. And so what you will notice, and for some reason my point is going away, here it comes, is that the red colour is a simulation for someone with a severe obstructive lung disease, whereas the purple colour and the green colour is for someone with a mild obstructive lung disease. And without going into the details, what you can clearly see is that where the particles deposit varies depending on the level of obstruction. So again, when considering the health effects of inhalation of particulates, the deposition of the particles themselves is affected by lung disease, which will therefore affect the way that the lung responds. So in short, the distribution of particles following inhalation into the lungs is heterogeneous and complex. So it's a complex area to try to unpack. So in considering that aspect of the health effects of particulate matter, the second thing to consider, once you've established, well, the particles are going to deposit in different areas of the lung, depending on a range of factors, including particle size and geometry and pre-existing lung disease. The question is, well, what happens to the particles when they're inhaled? And so what I have here is kind of a schematic of the different types of cells that particles might come into contact with. So this end on the left is basically the upper airway. So particles are being breathed in through here, and they will deposit at some point, depending on their size, bigger ones tending to deposit further up in the airway, smaller sizes tending to deposit further down in the lung. So when particles are inhaled, they will come into contact with the cells if they deposit, and a range of things can happen. The first thing that can happen is if they're deposited on these airway cells here, there's a mucus fluid layer that lines these airways, and if the particles become trapped in that mucus and fluid layer, these cilia in the epithelium will beat and move those particles back up into the upper airway, and you'll either cough them out or you'll swallow them. So that's the mucociliary escalator. That's the system that tends to remove particles or other non-host things, particles, once they come into contact with the airway. So that's one potential outcome for inhaled particles. The other potential outcome is that once they come into contact with the cells, they actually enter those cells. So there are specialized cells in the lungs, known as macrophages, which are designed to soak up things that are inhaled. So really they're there to deal with inhaled bacteria and viruses. So they engulf the bacteria, kill the bacteria, and then those bacteria are disposed of. But particles can do the same thing. So they can be engulfed by these macrophages, or they can enter some of these other cells. And there's evidence to suggest that the particles will directly enter the cells that line the airway, and therefore can have health effects by that route. If those particles are small enough, they may also be able to penetrate the lung tissue and enter the circulation. And this is thought to be one of the main reasons why there's a strong association between exposure to particulate matter and cardiovascular disease. Because those particles can actually physically enter the circulation, they can then exert their effects on the cardiovascular system. And finally, if they do manage to make it through the airway tissue and into the interstitium, which is basically the tissue on the other side, they can also enter the lymph system. So if they're able to enter the lymph system, they can engage with the immune cells and influence the immune response. So as you can see, again, where the particles go are likely to have a big impact on the response. And the physicochemical characteristics of the particles are going to determine whether they're excreted via the mucociliary escalator, whether they're taken up by cells, whether they are managed to cross the airway tissue barrier, whether they enter the circulation, whether they enter the lymphatic system. So again, when trying to understand the health effects of particulate matter, it's a really complex system. And so just to highlight the fact that particles do enter the circulation. So this is a study looking at nanoparticle deposition in a mouse model. So basically what they had was a set of radio-labeled particles. The mouse inhaled those particles, and this is a live imaging system where you can track where those particles go. And you can see over time that the particles deposit in the lung, and you eventually start to see those particles moving into the bladder. So these particles have made it all the way into the circulation and then are being excreted through the kidney into the bladder and will be excreted with the urine. And so they can have effects at any point of that process. So when they come into contact with the cells in the lung, when they come into contact with the cardiovascular system, and when they come into contact with the urinary system. And so one of the other complicating factors in all of this discussion is how long particles tend to actually stay in the lung once they're inhaled. And so this is a really interesting data set from the Rocky Flat Plains nuclear facility in the US that was shut down for a range of unfortunate reasons. And so what they did was they studied the deposition of radioactive plutonium oxide in workers and residents and looked at how long the particles were retained in the lung. And so the important data to look at is this data here on the left. And so what you can see is that even after 900 days post-exposure, there is still around 36% of the inhaled particles in the lung. So these particles stay in the lung for a long time. And so one of the interesting things is to consider, well, what happens to those particles? Does the system stop reacting to those particles? Does it continue to react? And perhaps this is an explanation for some of the things that you observe in, for example, silica-induced lung disease, where the particles are retained the system for a very long period of time. They're associated with the development of tissue injury, and that tissue injury forms scars and results in the severe lung disease associated with exposure to silica particles. So, particles can deposit in a range of areas in the lung depending on their particle size. Once they enter the lung, there's a range of fates which will impact on the potential health outcomes, and some particles, if they do remain in the lung, will stay there for a very long time and impact on the potential health consequences. So I don't expect you to pay too much attention to the detail here, but this is just to highlight the fact that inhalation of particles at the lung has the capacity to impact a range of adverse responses or cause a range of adverse responses across the body. So it was linked to effects on the cardiovascular system, the lung obviously, effects on the kidney, heart, and on the brain. And so what I'm going to do now is try and talk a little bit about some of the common mechanisms that are generally thought to drive some of these adverse responses. So when a particle comes into contact with tissue or a cell, whether or not there's a response and the nature of the response will depend on the properties of those particles. But in general, what's thought to happen with particulate matter once it comes into contact with the tissue is a series of responses that are probably driven by the innate response of the tissue or the cell to try to remove potentially damaging objects. So really what typically happens is that these particles stimulate the processes that are usually designed to kill bacteria and viruses. The problem with that is that those processes don't have any virus or bacteria to act on. So what those processes tend to do is to act on the healthy tissue that's surrounding the point where the particles have come into contact with. So it's triggering a series of responses that aren't necessary because these particles can't replicate and cause an issue, but that response can directly damage the tissue that it comes into contact with. So there are a range of processes that are thought to be involved, including oxidative stress. The particles could directly kill the cells. They could potentially alter the DNA or the genetic code and cause their effects. They can drive inflammation, and it's that inflammation that can then cause further tissue damage, or they could modulate the immune system. And the combination of these responses will determine whether or not there's local tissue injury. Now, most healthy individuals, if you're exposed to an acute particle exposure event, you'll have one of these responses and you'll recover. If that exposure event continues, or if you have a pre-existing disease, that could lead to permanent tissue injury, so scarring, modelling, or destruction of the lung tissue. If certain pathways are set off, it could promote pathways that are relevant in the development of tumours and cancer. Or if there's immune effects, it could impair the response to secondary insults. So rather than having a direct effect on the individual, it might be that the particulate matter alters your response to a secondary insult, such as a respiratory virus or something similar. Okay, so that's sort of the back of the envelope summary of the general mechanisms that cause the health effects associated with particulate matter, or what we know so far. I will mention oxidative stress in just a little bit more detail because it's talked about quite a lot in terms of the health impacts of particulate matter. It's certainly an important process. It's not the only process, but it's worth mentioning in a little bit of detail because of that. And so when considering oxidative stress, you need to consider two things, and that you need to consider the balance in the system between the production of antioxidants, which prevent oxidative stress, and the generation of reactive oxygen species, which is what promotes the oxidative stress. And at any given moment, if those two things are in balance, then the system is okay. It's when that system is thrown out of balance that there is a problem. Now, what I should point out that the oxidative stress response is actually a perfectly normal response to invading pathogens. So oxidative stress is actually an effective means of killing invading bacteria. The problem is, as I mentioned before, with particulate matter, is there's nothing to kill per se. And so what typically happens is that oxidative stress then causes damage to the surrounding otherwise healthy tissue. And it does that by directly causing tissue injury, or by promoting inflammation and recruiting other factors that then cause the tissue damage. So that's one aspect of the response. The other thing to consider, and I've mentioned this a number of times, is how the particle structure might actually impact on the response. So the particle structure, we know, as I've already mentioned, will impact on whether or on where the particles are deposited, but the particle structure can also affect the cell response. So we know that particle size determines where the particles go. I've mentioned that already, but they also determine how the cells respond. And so again, apologies for the excessive detail here. I just want to point out one thing. So this is a schematic of how a cell, a lung cell, responds when it comes into contact with silica. So we know that silica particles are highly detrimental to the lung, can cause a range of diseases, and is prominent in the thoughts of many at the moment because of the issue with silicosis associated with cutting Caesar stone. That's becoming a real problem in this space. Nonetheless, we know a lot about how silica affects the cell response. If you have large particles of silica, it tends to signal through this particular pathway here. This pathway causes the generation of inflammation, which perpetuates the response and can cause tissue injury. If your tissue comes into contact with a relatively small silica particle, that particle can then enter the cell and cause a similar response, but via just separate pathway. So I suppose what I'm trying to highlight here is that even though these two particles have exactly the same surface structure and chemical structure, the cell response will vary depending on the size of the particle. So particle size has an effect on the cell response. The surface structure can also determine how the cell responds. So again, using silica as an example. And so this is a cell study where they put different concentrations of two forms of silica. So these forms of silica have the exact same chemical structure. What differs is the surface structure. So amorphous silica tends to have a relatively smooth surface, whereas cristobalite tends to have a relatively jagged surface. And these surfaces, even though the cells are exposed to the same concentration of silica, so 150 micrograms per mil in this case, the response was vastly different. So the amorphous silica had no effect on the inflammatory response. So the surface structure also affects how the cells respond. So far I've talked about how particle size can affect the response by potentially signaling through different pathways in the cell. I've talked about how the surface structure also impacts on the response. So what about the chemistry? And so I'm going to take two extreme examples here, primarily because we know the most about these two particles and their health effects, particularly the cell interactions in the lungs. So if we take diesel exhaust particles as the classic urban-derived particulate matter that has been well established as having adverse health effects across the body systems, and silica on the right, and think about how the chemistry of those two individual particles might affect the response. So diesel particles are quite a complex particle. So they typically consist of a carbon core with a range of chemical compounds absorbed onto the surface of that carbon core. So here we have them illustrated in the little yellow hexagons there as organic compounds. There are also transition metals absorbed onto the surface of these particles. And what you'll see is that, based on this schematic, again, details unimportant, is that the organic compounds and the compounds that are on the surface of the diesel exhaust particles can stimulate a whole range of receptors that sit on the cell surface, cause a signaling cascade, which then leads to an inflammatory response. Okay, so part of the diesel exhaust response is probably driven by the fact that the chemical compounds that are absorbed onto the surface of the carbon stimulate the activity of these receptors and cause inflammation. So part of the diesel response is probably explained by this process. If you take particles in general, there are a range of receptors that sit on the surface that will respond to those particles, irrespective of the chemistry. So there are innate damage response cell receptors, which are specifically there to detect an impending threat to the system and to respond. So particles are well known to stimulate those pathways, and it's likely that silica acts through this sort of mechanism, but also diesel is likely to stimulate this mechanism. So it's entirely possible that one of the reasons why diesel exhaust particles are such a potent pro-inflammatory particle and why they are so strongly associated with the health effects of particulate matter is that they can stimulate a whole series of different pathways that promote inflammation and tissue injury. But hopefully what you can see, even from this slightly complex diagram, is that the chemistry of the particles is also going to vastly affect the response, which will explain why there are significant variations in the inflammatory response depending on which particles you look at. So we know that inflammation is important in potentially driving the adverse health effects associated with exposure to particulate matter. So let's explore that a little bit further. And so I'm going to talk through a series of examples, primarily from my lab, where we've tried to look at this exact phenomenon and try to understand how particles from different sources might impact on the response of the lung in particular, and therefore impact on the health risks. So this is a very simple diagram here showing how the inflammatory response, so this is in an experimental mouse model system where we expose mice to either diesel exhaust particles or particles from a geogenic source. And by geogenic, I basically mean crustal or earth-derived particles, so wind-blown dust. And what you can see immediately from this figure here is that the geogenic particles on this measure of inflammation had a much bigger effect, and on this measure of inflammation here also had a much bigger effect. Now you might argue from that simple bit of data that, well, it seems as though these crustal or geogenic particles are more pro-inflammatory. However, as with everything, the story's not that simple. And if you look at other measures of inflammation, so on the left here, again, we have diesel. And so this is another measure of the inflammatory response measured over time as opposed to a single time point here. And what you see is that the diesel exhaust particles cause a big influx of cells into the lung 6 hours after exposure, and then that response dissipates over time. This is the same measure in the geogenic particles. My apologies that it's formatted in a slightly different way. And in this instance, the key bar to look at here is the dark bar. So these are the geogenic particles, and what you can see compared to control is that, if anything, the levels of these particular cells, which increase in diesel, actually decrease. So what you can clearly see from this data is that why you can't say whether or not exposure to geogenic particles or diesel exhaust particles is going to be worse for your health. What you can say is that the response clearly varies. And if that response varies, that means that the health impacts probably vary as well. So I'm going to unpack the geogenic or crustal example a little bit more. And so this slide is just to highlight the fact that there are populations around the world where exposure to these sorts of particles is high. So here we have, again, the skyline of London with Saharan blown dust, arid environments, and some data showing that these levels of particulates can be very high in certain regions. So in order to explore this, and so in this instance, this study was aimed at looking within geogenic particulate matter. So we know that wind-blown dust, the physical and chemical characteristics of wind-blown dust is going to vary depending on where those particles come from. So this study was based in WA, which is where I'm originally from. And what we did is we collected these particles from across Western Australia at 8 different sites. We exposed mice to the particles and then measured a range of outcomes. So what's important to note is of these 8 samples, we had a range of physical and chemical characteristics. So we have, for example, we had measures of endotoxin, which is a measure of bacterial contamination. We had measures of particle size, which varied, and we had measures of chemistry. So, for example, the silica content, the iron content, and the aluminium content. And we measured a range of things. So we measured inflammatory responses, and we measured the lung function response. And so with some sophisticated mathematical modeling, what we did was try to look at how the complexity or the variation in these physicochemical characteristics impacted on these outcomes. So I'm just going to show you one of those, one outcome from that analysis. And in this analysis, we're looking at lung function. So what we found in general was that these particles impaired lung function. So if you have impaired lung function, that's going to mean that you have increased risk or likelihood of being hospitalized for respiratory disease. So this is an important outcome. And we found 2 key observations. Firstly, the particle composition, that is the chemistry of the particles, impacted on the response. So intriguingly, in this particular data set, we found the higher the iron concentration, the greater the impairment in lung function, whereas the higher the silica content, the lower the impairment in lung function. Again, highlighting the effect that the particle composition affects the response. We also found that particle size affected the response. So while the median particle size had no effect on the response, the variation in the particle size certainly did. So the more variable the particle size was in the sample that we used, the greater the impairment in lung function. So again, highlighting this idea that both the chemistry of the particles and the physical characteristics, in this case the variation in particle size, can in fact impact on the response. So just to highlight this further, another study we collected 36 dust samples from the roof spaces of some suburban homes. And what we did was we exposed a cell line. So these are studies in a dish where we've taken lung cells, grown them up, and we exposed the cells to the particles and then measured the response. So again, we characterized the particles. This time we did a more comprehensive analysis. So we assessed them for polyaromatic hydrocarbons. We assessed them for 22 different elements, endotoxin and particle size again. And we just primarily measured two key outcomes. One, did the particles kill the cells? And two, did they produce an inflammatory response? So again, without going into the intricacies of the study or the analysis that we did, the problem that we had with this data set is we had a whole range of measures of the physicochemical characteristics of the particles. And these particles, these chemical characteristics are likely to be highly correlated with each other in some instances. So what we did was a modeling analysis where we tried to look at collections of characteristics that were related to the response. And what we found was that, as you would anticipate, the bacterial contamination of the particles affected the response. So the higher the bacterial contamination, the more inflammation. We also found that this particular outcome, now this outcome is primarily linked to combustion-derived particles. So the higher the concentration of combustion-derived particles in the samples, the greater the inflammatory response. But in this instance, we found that particle size had no effect. So in this system, using these kinds of cells, we found that the chemistry had a big impact, whereas the particle size didn't have an impact. In contrast to the previous study where we had wind-blown dust particles or crustal particles, particle size certainly had a big impact. So just to add to the complexity of the discussion. All right, so up to this point, I've primarily talked about the healthy response. That is, so what happens to the cells or what happens to the organ system when a particle happens to come into contact with those cells or that organ system. What's also important to consider, as I mentioned earlier, is how disease might affect the response. So I've already mentioned the fact that we know that disease probably impacts on the way that particles are deposited in the lung. There are some, I just want to talk briefly about some human exposure trials. These studies fascinate me for a number of reasons, primarily the ethical implications of essentially putting someone in an exposure chamber and exposing them to exhaust from a truck. Nonetheless, these studies have been done and they have some fascinating results. So in one study, what they did was they exposed otherwise healthy individuals to diesel exhaust and found that when they exposed them to diesel compared to control air conditions, there was an increase in the inflammation and no change in lung function. So in this instance, at the doses that they used, they found that the exposure to diesel exhaust particles caused an increase in inflammation, which probably explains part of the adverse health effects of diesel, but in this instance had no effect on the lung function, so their capacity to get air in and out of the lungs. Another study, very similar design, rather than using healthy individuals, took asthmatic individuals. And what was fascinating about this particular study is that in asthmatics, they found no effect on inflammation, but a clear effect on lung function. So this is a measure of how readily the airways constrict, where a lower number here means a higher likelihood of constriction. That is, they required a lower dose of this compound here to cause the airways to constrict. So what they found in the asthmatic individuals is that there was no change in inflammation, but exposure to the diesel exhaust particles clearly dropped lung function. or cause a reduction in lung function. So here we have an example where the disease clearly impacts on the response. So what about the effect on the disease response? So that's how the disease affects the response to particles. What about the effect of particles on the disease response itself? So this is another study that we did using some of the geogenic or crustal particles that I talked about earlier. But in this instance, we were looking at a study of how those particles affect the response to influenza. And so what we found was that particles, depending on which region they were from, which affects their chemistry, had an impact on the response to influenza infection. So particles from this particular location caused an increase in the amount of influenza that replicated in the lung. And we know that the more that the influenza replicates, the greater the inflammatory response. And what we found was that the magnitude of this response was linked to the ability of the lung to produce this particular mediator here, which is known to be important in clearing the virus. So basically what that suggests is that inhalation of these particles impairs the viral response, which causes increased production of the virus in the cells, which is likely to lead to a greater impact on lung health. And so just to highlight this point that particle characteristics have an impact, we did a similar analysis where we looked at the physical and chemical characteristics of the particles. And what we found, again, intriguingly, that iron seemed to be the most important chemical in these particles. The higher the iron content, the lower the production of this mediator, and the higher the viral titer. That is, in particles that had a relatively high iron content, the system had an inability to effectively clear the virus. So the crustal PM exacerbated the response to respiratory infection, and this was positively associated with the iron content of the particles. So here we have, in the previous slide, I talked about evidence of how disease affects the response to particulate matter, and here we have evidence of how particulate matter might affect the disease response itself. Okay, I'm getting towards the end now, and so I just want to talk about one last aspect that I haven't touched on. So in the previous slides, I talked about the effect of disease on the response to particulate matter and the effect of particulate matter on the response to disease. What I haven't really talked about is the evidence for particulate matter actually causing disease. So there's lots of evidence now to suggest that exposure to particulate matter, particularly in early life, increases the risk of a range of diseases, particularly lung disease and asthma, including asthma. So in this study, again, this is a mouse study, and what we did here was we took particles sampled, again, from suburban homes, and we exposed the mice in utero. So the pregnant mice were exposed to the particles, and then we examined the outcomes in the offspring. So these are measures of outcomes in the offspring when they're 2 weeks of age, which in human terms is roughly equivalent to pre-teenager. And what we found was several things. Firstly, we found that if the mice were exposed in utero to the particles, they had a reduction in body growth. They had an impairment on lung mechanics. And so on this scale here, the higher the value, the worse the lung function. So they had an impairment in lung function, which seemed to vary depending on where the particles were collected from. So in this particular example, this sample 2 showed an impairment in lung function, whereas these other samples didn't. And on top of that, the mice had a reduction in this particular subset of immune cells, and these immune cells are known as regulatory T cells, and they're very important in mediating the response to infection. Mediating the allergic response, I should say. So if you have a lower number of these cells, you're more likely to have an allergic response. So in this very simple experiment, what we've shown is several things. Firstly, that in utero exposure to particulates can reduce body growth, can reduce postnatal lung function, and can modify the immune response. So collectively, these observations probably explain at least some of the mechanisms as to why there's an association between early life exposure to particulate matter and epidemiological studies, and the increased risk of lung diseases, particularly allergic lung diseases like asthma. So it's been a bit of a whirlwind tour. I'm just going to try to summarise all that now. And so overall, the health effects of exposure to air pollution are diverse, and the health effects of air pollution are driven by the interplay between the pollution composition itself, the nature of the exposure, and the subject, whether they're healthy, aged, young, or diseased. And the interaction between all of those things determines whether or not there is a response and what that response is. The mechanisms underlying the adverse health effects are highly dependent on the physicochemical characteristics of the particulate matter, because those characteristics impact on where the particles are deposited. It impacts on how the cell responds to those particles, and which therefore impacts on how the overall system responds to that and whether or not there's an adverse health outcome. The health effects of air pollution are driven by oxidative and non-oxidative responses that induce tissue damage and promote inflammation, and whether or not there's a health outcome, that is an increased risk of hospitalisation or an increased risk of disease, will depend on how severe those responses are. And there's clear evidence that disease itself modifies the response to particulate matter, is exacerbated by exposure to particulate matter, and can be caused by exposure to particulate matter. And on top of that, the physicochemical characteristics of the particles will impact on those responses. So I suppose the take-home message is that particulate matter is clearly linked to a range of adverse health outcomes, and the complexity and the response is highly complex and depends on a range of factors. So when you're considering trying to mitigate against the health impacts of particulate matter, you need to consider all those factors. And so applying single measures of particulate matter exposure, for example, to try and mitigate against the health impacts might underestimate the complexity of the situation and ignore the fact that the chemistry and the nature of the particles has a significant impact on the response. Thank you very much for your attention. Happy to take questions.
Okay, thanks for that, Graeme. That was really, that covered a lot of ground in that time. Thank you so much. Can I ask people if they have questions to type into the chat box, please? It's probably the easiest way to post questions for Graeme.
So how do I see the chat box?
I'm not sure what screen you're seeing. I can read them out if you can't find it.
Oh, okay, no, I found that now. Thank you.
You did find it? Okay, any questions coming through from anybody? Can you say that question there from Anthony?
I can. The implications, I suppose, were one of the challenges when trying to understand safe limits of particulate matter exposure, or what's a practical level to set, is that particle size isn't the only thing that needs to be taken into consideration. The other thing that needs to be taken into consideration is the chemistry of the particles. And there's emerging data that there is probably no safe level of particulate matter per se, but when setting limits in different areas or different regions, the other thing that needs to be considered is the primary source of the particles, because we know that the primary source of the chemistry of those particles will significantly impact on the response. I can see a question from Michael. Thanks, Michael. The one thing that I neglected to mention in that particular study that slightly complicates the interpretation is that those asthmatics were all on inhaled corticosteroids. So that probably masked the inflammatory response. It's likely that in that particular situation, I suspect it's a direct effect on the epithelium, and it may well be that the particles reduced the effectiveness of the barrier of the epithelial cells, which meant that the methacholine actually had more access to the airway smooth muscle. But that's that's pure speculation at this point. Okay. That's an interesting question, Jeff. My comment to Jeff's question about why the epi study is relatively consistent between various locations. I'm not sure that the epi data on the geogenic particles, for example, is there. And so what I actually suspect is that the consistency of the response is because most of the exposure in most population is from combustion-derived particles. And while I focused on diesel exhaust particles, a lot of combustion-derived particles will have a similar structure with a carbon core and adsorbed organic compounds and transition metals. So it's likely that the bulk of the epi studies are probably examining a similar exposure. I suppose my comment is more around extremes of exposure. So, a city versus a regional town, for example. Okay. So the comment here about whether or not exercise affects the degree of inhalation and therefore the effects, and I think the short answer is yes, but the data would suggest that so I suppose one of the thoughts was that, well, what's the balance of risks between going out and running down a busy street and being exposed to particulate matter versus the effects of exercise? And I think the data would suggest that the health effects of the exercise far outweigh the potential health effects of exposure to particulate matter. But there is evidence that exercise will increase inhalation of particulate matter and therefore potentially increase the risk of adverse health effects. So that's an interesting question. So that's true about being exposed to smoke and salt spray and volcano emissions. The salt spray one's an interesting one. I'm not aware of much data suggesting that there are health effects of salt spray. One of the interesting things about salt spray, I suppose, is that many of the chemicals in that sort of particle are in the lungs itself. So in some ways, that's not they're probably not a foreign body response. The smoke one is interesting. Yes, it's true that from an evolutionary point of view, humans have been exposed to a lot of smoke, but I suspect that the nature and the intensity of the smoke has varied. There's a big difference between an open fire in the outdoors versus a smoke that's being generated indoors at a highly concentrated level. And so, yes, there are. It's likely that we've co-evolved, I suppose, in that exposure. But I'm not aware of any data that suggests or evidence to suggest that there is a lower limit of an effect of particulate matter. But that's an excellent question and something I hadn't really considered much previously. How are we going? Okay, so the question here is on a carbon-related comment. What if anything makes this? I'm not sure that the evidence suggests that diesel is worse. It's just that diesels are so much have been the subject of so much study. So there's so much data on the health effects of diesel. I would anticipate that many of the health effects of other combustion-derived particles would be similar, but the data just isn't there. So one of the things that I'm quite interested in at the moment is the effect of particulate matter from landscape fires. So, what would be interesting would be to do a side-by-side comparison of diesel exhaust particles and combustion-derived particles from a different combustion source. But that's an excellent question. And it may well be, you're right in that last comment there, that it will vary depending on what sort of organic compounds are absorbed onto the PM. Thanks, Rachel. Interesting question. Based on our data, so one of the things that I should say that there are a few studies that have looked at this, and it's certainly an approach that we haven't taken. What we've tended to do so far is to take real-world particles and then try and model the response on the basis of variation in the physical and chemical characteristics. We have done a few studies where we've taken relatively pure particles, so for example, iron oxide and silica particles, and looked to see whether there's an interactive or synergistic effect. In that particular scenario, we didn't find a synergistic effect, but that doesn't mean that it doesn't exist in other scenarios. I think, because of the complexity of the responses, it gets very difficult to run those studies, but it's certainly something I'm particularly interested in. That's an interesting question. It could be nonlinear. In our systems, I haven't. I mean, given in any of the experiments that we've done, we haven't observed any non-linearities, and I'm not aware of. I'm probably not across the intricacies of the epidemiological data to comment on that. It's certainly possible. If you look at the toxicological models of how particles interact with cells and their trafficking, given the complexity, it's highly likely that it's nonlinear, but I'm not aware of any data to support that. One of the time question is actually really interesting to me. One of the things that I don't think we have a handle on is why the system seems to switch off. So one of the data sets that I didn't show you, and it's kind of linked to this idea that insoluble large particles tend to be retained in the lung for a long period of time, is why the system doesn't tend to continue to respond to those particles. And we have some evidence that, at least in the medium term, that the inflammatory response gets dampened following an acute exposure to particles. So what you typically see in our model systems is an increase in inflammation that then resolves after about 24 to 48 hours. But if you monitor the system for longer periods, say 7 days post-exposure, what you tend to see is a reduction in the inflammatory response below baseline, which would suggest that there's actually an active process that dampens the inflammatory response, which might explain why the inflammation doesn't continue to, because otherwise you would anticipate on the basis of the fact that the particles stay in the lung for a really long time, that the inflammatory response would just continue and in perpetuity over time while those particles are there, but it clearly doesn't happen. Interesting question. I'm not aware of the immune data on healthy individuals. We do know that particles will modulate the immune response, particularly diesel exhaust particles. Again, not because they're anything special, but that's because we have the data, because that's where the data is. Because diesel particles are such a complex particle, they have a range of, they stimulate a range of pathways, and one of them is certainly the immune system. So diesel exhaust particles are a really potent adjuvant for allergic responses, so they will promote allergic responses. And that will be in healthy individuals as well. That's a great question about whether or not there's enough data yet. I think the short answer is no. And I think where the big gap in the data is with the crustal particles, and the reason that there's lack of data in that space is because they're typically challenging population data to get, because unlike a large city where you have relatively high numbers and relatively high numbers of events, the communities that tend to be exposed to high levels of geogenic particles are remote or have populations that are relatively low, such that it's really hard to determine what the dose response is for exposure to those particles. So I think the short answer is no, and then more work certainly needs to be done in that space. There you go. Interesting. Yeah, that's a good point, Jeff, about only recently being measured. That's an interesting point. I mean, yeah, you would anticipate that exposure to smoke has been around for a really long time. I think what would be interesting to know is whether or not the exposure levels have been consistent over time. And what the evolution, and what the evolutionary impacts of that were, or was there enough selective pressure in response to smoke. So were the health effects of smoke sufficient to prevent, were the health effects so severe that it prevented, had reproductive outcomes that would affect growth or affect the likelihood of reproduction? Or are the health effects of smoke cumulative such that they're only observed in old age, and it's the fact that we tend to live longer these days, and the biggest signal in the effects of smoke is probably in chronic obstructive pulmonary disease, which is typically a disease of middle to late age. So perhaps it's the evolutionary pressure was there, but it wasn't severe enough to impact on, a selective process that would modify the cellular response. Yeah, so I think that question about updating guidelines is really interesting. I think we don't have the data. I think there's good epi-dose response curves for urban populations. I don't think we necessarily have that data for non-urban populations, and I think that's where the work needs to be done to inform those guidelines. And I suppose my work is often quite distant to actually setting a number, because most of my work is in experimental studies. But what my work can do is inform how you might design an epidemiological study or a population study that would answer some of those questions. What is the dose response for particles from different exposures? Excellent, and I appreciate all the thanks, by the way.
I think that brings us to the end of the webinar. Thanks very much, Graeme, for such a great presentation, and thank you to you all who are still there for tuning in today. And yeah, that brings us to the end. So thanks very much, everybody.
Thank you. And thanks for taking time. I would be happy to take questions offline if anyone has any burning questions as well. Thank you.
Okay, thank you.
Some of them are for